Now, we said that density equals mass over volume, and if you want to take out the result of the mass, we're going to multiply the density by the volume. Now, in this question, he wants the mass, okay? So, we're going to take the density that he said, a width density of 0.136, and we're going to multiply it by 10 to the power of 6. Do you know why? We're gonna turn it, okay, from centimeter to meter. Okay? That's why we multiplied it by 10 to the power of 6. And we're gonna do like the multiplication, and the result will be 6.48 by 10 to the power of 8 gram. Okay? Now we have the note. Note we converted the volume into centimeters square to the power of 3 because our density was in gram to the centimeter to the power of 3. We have to match the units. Okay? It's very important to match the units because you can't do them with the one in centimeter and one in meter and one in the negative force and not the positive force. The International System of Units is called SI. The most commonly used are the one in the red triangle, which is a liter, a kilogram, a second. You have an ampere, a Kelvin. Ampere is for electricity. Kelvin is for heat. Like when you say Celsius, you have also Kelvin. You have candela and you have mole. Okay. This is like the international use. It's called SI, an acronym for System of International System of Units. Here we have the general size of living things. You can see the smallest one starts from 0.1 nanometer, which is an atom, and it grows to the great blue whale, which is over 1 kilometer. You have the range from 0.1 nanometer to the 100 micrometer. These are seen in the electron microscope. One of the most intricate microscopes that were invented is the electron. And you can see from the range from 100 nanometer to 100 centimeter, it can be seen with light microscope. A light microscope can't see every little thing, but you can see a virus with a light microscope, but you can see a human egg or a frog egg with a light normal microscope. From 1 centimeter to over 1 kilometer can be seen with the human eye. Like you don't need a microscope or any devices like the mouse, an ostrich egg, or of course a human. Now we have each unit with what they equal. So we have a kilometer is 10 to the first of 3 meter, or we can say 1,000 meter. Each kilometer is 1,000 meter. A meter is 100 meter, of course, you know. A decimeter is 10 to the first of minus 1. It means 0.01 meter. Okay? And so on. So when you have an equation, let's say it's in a meter, and you have to change it into a kilometer, you have to take 1/3. Okay, so that's 10 to the force of 2. We have here the red blood cells diameters. It starts from the goat, which is, of course, in this unit we're going to use micrometer. So we have the goat, which is 3.9 micrometer. Of course, this can be seen with a light microscope, but it has to be seen with an electric microscope. Then you have the horse, which is a bit bigger. We're going to move to squirrel, 5.6. Human, the human RBC is 7.9 micrometer. Indian elephant, hummingbird. Hummingbird is a type of bird, of course. A chicken, a salmon, which is a type of fish, crocodile, and the biggest RBC you can see in nature is the 3-toed amphibian. It's like this one, the last picture. We're going to go back to the equations at the end. You have here the T4 bacteriophage. Okay? This was seen, of course, in an electric microscope. These are the parts of the T4 phage or the T4 bacterial phage, and each of these measurements. Okay, the numbers and letters refer to the constituent protein. Okay, it means how much protein in each part has. You can see the parts, like we call this part the head. Okay, this is the tail or the tube, and these are the basal plate hubs, and these are the base plate wedge. Okay, and these parts are the fibrinogen. These appendages are used to inject into the creature, like this one. Okay, and then the bacteriophage will insert its genetic material into the human that will be engaged in. This entire creature is only 100 nanometer. Okay. Um, we're going to move on to measurements in general. Measurements and uncertainty. Most experiments require scientists to make measurements. Every experience that you do, you have to make measurements to take out the correct result. Measurements are rarely exactly the same. It means when you have an experiment in the lab and you make measurements, they usually are exact in the numbers. Okay. Measurements are always somewhat different from true value. We can't have all measurements true. Okay, sometimes there are some error in it. Okay, the deviations from true values are called errors. So when we have measurement and we have a result, it can't always be 100% true. There can be some mistakes in it, and these mistakes we call them errors. Source of errors. Generally, in experiments, we have two sources of errors in measurements, which are limitations and the sensitivity of instruments. It means maybe the equipments are not new, are not updated. Maybe there are some mistakes in the programming of these instruments, so they are not as sensitive in taking out the actual result. And the second error is imperfections in experimental design or measurement techniques. Of course, as humans, we may, as like it's normal to make mistakes as humans. So maybe in measurements there may be plus minus of what you add or what you remove. So these are the most common two errors of measurement. Errors are often classified as random and systematic, and we're going to find out what each means. First, we're going to take random errors. Random errors are always present in the experiment, okay? And they are shown, measurements are often shown as, do you see this graph? You have here the frequency, and you have here the average, or the y-axis, or the x-axis. You have the green line, which is the distribution of a random error, with the blue line with the distribution of an x with no random error. So you can see in this diagram that the distribution of random error are more frequent because the peak hits with the frequency higher. The distribution of random error are always present or more often happening than the other type of error. Now, we have the sources. You can have operator sources and changes in experimental conditions. Operator errors, it means with the human itself. When they make an experiment, They might have like an accident, some faulty measurements, something wrong with the equipment. It has a lot to do with with the experimental herself or herself. The second one is changes in experimental conditions. Maybe the equipment or the materials we use in the experiment are affected by heat, affected by light, maybe by temperature. So there might be some changes in it. So this also calls for errors in the experiment. Now, how do we minimize random errors? We take repeated measurements and we calculate their average. Maybe we take 5 or 6 measurements of the same experiment in the lab, and then we add them to each other and divide them on their sum, and this is how we take out their average. Now, systematic errors, they are typically present. It means not always. They might happen and they might not happen. Okay? The measurements are given as we're going to look at the same diagram that we saw before previously, the frequency to the x-axis. We have the red line is the distribution of x with systematic error, and the blue line is the distribution of x with no systematic error. Notice that systematic error does affect the average. We call this a bias. Okay? The sources are instrumental, physical, and human limitations. As we said before, maybe the equipments are not new, are not loaded, are not kept up with, they are not maintained. So this also may call to systematic error. Like this one, a device is out of calibration. How do we minimize them? We have to be careful with our calibration, and we use the best possible techniques. Okay, maybe for equipment you buy the best one or the best company, the recommended or whatever. Now we're going to learn about the difference between precision and accuracy in measurements. Precision means how reproducible are measurements. However, accuracy means how close are the measurements to the true value. Okay? So when someone says accurate, it means he is as close as possible to the real result. But when he's precise, he could be he could be on a true result, but not as close to the real result. There is some type of analogy. It's called the dartboard analogy. This will teach you the difference between the accurate and the accuracy and precision. Imagine a person throwing darts, trying to hit the bullseye. The bullseye is the ball, the star, the circle in the middle. In the first picture, the first one on the left, you can see how the bullets are scattered all over from the outside. They are not accurate nor precise. Okay? Because look how they are scattered in no particular pattern. The second one from the left is accurate and not precise. All of these scores or darts as close as possible to the bull's eye, okay, but they are not precise. They are scattered. The third one is not accurate, but they are precise. Okay, they are not close to bull's eye. However, they are organized in their scattering. The fourth one, they're accurate and they are precise because they hit bull's eye and they're organized in their scattering. Okay, so this basically tells you the difference between the precision and accuracy. Precision of a measurement. You can look at this ruler, okay? You can predict what the arrow is pointing at. We have 26 centimeter, point something, okay? So when you look at this ruler, maybe you will estimate like 0.1 or 0.2, Okay, this is called precision. You're not correct on the actual point of it, but you're close. Okay, so you can see here measurement equals or almost 26.13 centimeters. Now, of course, when we look at this, we know it's 26.1, but we don't know what comes after in these numbers. So, when we say it's 26.1 centimeters, this is a precision dot. The last digit is called an estimate. Okay, we predict it. It's not 100% sure, but we predict it. And this precision is limited by the instrument, by the ruler that we just used. Scientific data. Scientists always want the most precise and accurate experimental data because accurate by itself may cause some errors later on, and precision by itself will also cause errors by itself later on in the experiment. So, the precision and accuracy are limited by the instrumentation and data gathering techniques. Okay, like like this ruler, it may not give you the most accurate numbers. It will help you with precision, but you only have this ruler, so you are limited by the instrument. You don't have any further instrument to show you an accurate result. This is what you have, so we are limited by it. And there's a lot of experiments, especially when we do them in underdeveloped labs. Okay, they don't give us as accurate and precise results because we are limited by the instruments. The more developed, the more new, the more maintained instruments and equipment we have, the more accurate and precise results that we'll see. How do we deal with errors? We have to identify them and their magnitude. Okay, we have to first know where we made a mistake or where we made an error. We have to identify them. And then we try to reduce it. How do we reduce the error? We reduce the error by using better experiments and better experimental design, and we have to collect a lot of data. As we mentioned before, we have to use or we do more than one experiment and collect all of the measurements, and then we take their average to have a real or a precise and an accurate result. Okay. We're going to do now some calculations. Okay. Filling of the heart occurs during diastole. Diastole means when the heart muscle relaxes. You know when the heart pumps like this? So in the state of relaxation, like this, it's called a diastole. Okay? The maximum volume of blood within the ventricles of heart at the end of the diastole is called the end diastolic volume, EDV. So when the heart starts pumping blood and it reaches the point of relaxation, the maximum volume of a blood that enters the ventricles within the state of relaxation is called EDV. After that, the heart will contract like this. So the contraction of heart is systole, and the volume of blood remaining with the ventricle after systole is called end systolic volume. So the heart pumps blood in the state of relaxation is called diastole, and the blood flow, the maximum rate of that flow in that state is called ATP. However, when the heart contracts and the blood, there's for like a momentum of a second, a blood remains in the ventricles during the contraction of the heart. This is called ESV. Okay, it's called the end systolic volume. Strong volume is the volume pumped by the ventricles. It means the blood in general that goes in and out of the heart during the action of pumping. How do we know the mean of stroke volume? You're gonna say ETV minus ESV and diastolic volume minus and systolic volume. Stroke volume is also related to cardiac output. So whenever you see the expression cardiac output, it has the same meaning as stroke volume. Cardiac output, CO, is the volume of blood pumped per minute, and heart rate, HR. We measure the heart rate by using BPM, which means beat per minute. So whenever there's a stroke volume, there's also connected CO, which is a cardiac output. You can also know the mean of stroke volume by dividing the cardiac output over the heart rate. So these are two ways to know how to know the result of the stroke volume. We have the first equation. If the left ventricle of an individual has an ETV of 140 milliliters and an ESV of 50 milliliters, then calculate the stroke volume. Okay, so what do you say? We're gonna say ETV minus ESV, so it's 140 milliliters minus 50 milliliters, and the result will be 90 milliliters. Okay. The second one is kind of tricky. The ejection fraction is the percentage of blood volume ejected with each beat. What is the ejection fraction of the left ventricle in question 1? Okay. So we have the ejection fraction is the percentage of the blood ejected with each beat. Remember when we said the stroke volume equals the cardiac output? Okay. So ejection fraction also equals